Hi, I'd like to take this opportunity to introduce you to a recent paper that's appeared in Biochemistry. It's on the analysis of Streptomyces coelicolor phosphopantetienyl transferase, better known as an acyl carrier protein synthase or simply as ACPS. My name's Chris Arthur, and the work presented in the paper is a combination of largely Patrick Delalio's and my own work. The project itself grew out of a side project I took on whilst I was undertaking a postdoctoral placement with Drs. Matthew Crump and John Crosby at the University of Bristol in the UK. The side project that I decided to take on was to attempt to crystallise a protein that I was working on in the lab, this ACPS protein. So luck would have it, I managed to get crystals of the ACPS, and that allowed us to extend an existing collaboration we had with a group of Andrea Hadfield, and a lot of the crystallography work was then taken over by her PhD student at the time, Patrick Delalio. So the first question you're going to ask me is, what is a phosphopantetienyl transferase? Well, this protein here is an acyl carrier protein. Acyl carrier proteins are incredibly common in all cell types. Okay. Their role is to transfer intermediates in biosynthetic pathways. They're most typically found in pathways for fatty acid biosynthesis, polyketide biosynthesis, and the related peptidyl carrier proteins are also found in non-ribosomal peptide synthesis. Now, the role of this ACP is to shuttle intermediates between the enzymes that actually generate the small molecule that we're interested in, whether that's a fat, a polyketide, or a non-ribosomal peptide. Now, the trouble is this protein itself can't do the job. It must first be activated in order for it to perform its role in shuttling intermediates. And to activate it, we've got to add what's called a phosphopantethyne chain onto this protein. Okay? The phosphopantethyne is going to act as the point of attachment for our intermediates in biosynthesis, and also serve as a long linker that we can access active sites with. Okay? So we need a way of introducing this phosphopantethyne chain onto this acyl carrier protein. And that's the role of these phosphopantetienyl transferases. Okay. Phosphopantetienyl transferases, like ACPS, take coenzyme A and transfer it onto, or transfer the phosphopantethyne group, so this group on the left-hand side, this part of the coenzyme A, onto a conserved serine residue on an acyl carrier protein. And I'm just showing the ACP, that's just this circle here. Okay, so we take the apoform of this ACP, we take ACPS, and in a magnesium-dependent reaction, ACPS takes this phosphopantethyne moiety and transfers it onto the acyl carrier protein. Okay, so that generates our holo, or active, acyl carrier protein. So here's our ACP on the right, serine attachment site, and our phosphopantethyne chain. Okay. So that's the role of ACPS. Now, this process is biochemically very important. Without it, things like fatty acid biosynthesis, which we all require for our cellular function, and all cells require, couldn't occur. There is another reason for our interest. We have an interest in understanding the biosynthesis of fatty acids and polyketides. And what we can use this acyl carrier protein synthase for is for introducing new compounds into the biosynthetic pathway. How does that work? Well, on the terminus of the coenzyme A, there's typically just a thiol. Okay, and this thiol acts as the attachment site for the biosynthetic process that's occurring. What we can do, though, is we can use ACPS, or phosphopantethenyl transferase family in general, and these have very broad substrate specificity. They'll take practically any coenzyme A with anything attached onto this terminus here, okay, and transfer that directly onto an ACP. Why would you want to do that? Well, imagine, for instance, that we're studying polyketide biosynthesis. Now, you may not have met polyketides before, but they make up a vast array of our antibiotics, for instance, the tetracycline antibiotics. And a lot of the drugs that we use are derived from polyketides, or are polyketides themselves. So you can imagine if we can interfere with their biosynthesis, for instance, by including an unusual precursor, that we'd be able to generate new bioactive molecules. Okay. We can also use this function to introduce groups so that we can perform a number of biochemical assays to better understand how these biosynthetic pathways, whether it's fatty acid or polyketide biosynthesis, work. Okay. So it's incredibly important in terms of how we approach our understanding of polyketide and fatty acid biosynthesis. The thing is although this works and although it's a routine experiment in our lab, there's not much information on actually how this process, or why an ACPS will transfer a huge range of coenzyme A's. There's another side to this in terms of ACPS promiscuity. Not only are we interested in what sort of acylated coenzyme A's we're interested in, but there's also ACPS will also work with a huge variety of different ACPs. Okay. So we've been able to, for instance, use the ACPS that we're working on in this paper, which comes from soil bacterium, Streptomyces coelicolor, and use it to modify not only its natural fatty acid ACP, but also a polyketide ACP from the same species, polyketide ACPs from other species of bacteria, and even a fatty acid ACP domain from the mammalian system from rats. Okay, so a huge broad range of specificity in terms of the ACPs that we'll be able to use as substrates, and also in terms of what coenzyme A's we can use as substrates. Now, the project itself started with me attempting to crystallize acyl carrier protein synthase. That was the first crystal that we managed to get, but here we're presenting ACPS in complex with coenzyme A. Now, ACPS adopts this strange trimeric arrangement of proteins, so it's a trimeric quaternary structure. Okay, and each of these coloured proteins here is a separate ACPS molecule. Okay. The active site for ACPS isn't located on one particular protein, but is in fact spread across the interface of two different subunits of our ACPS trimer. So if we look at this ACPS active site here, you'll see that the coenzyme A binds not only to subunit C, but also in part to subunit A. The mechanism is magnesium dependent, and here's our magnesium cation shown as a grey sphere here. Okay, so we have 3 ACPS molecules, and those 3 ACPS molecules will bind 3 coenzyme A molecules, but the active site in coenzyme A will bind across 2 different subunits. A very unusual arrangement. Here's having a look at that active site in a little bit more detail here. Here we find the adenine ring of our coenzyme A coming up. Here's our phosphodiester that's chelating to this magnesium, so binding to that magnesium in the active site. Okay, we'll come back to that shortly. And here's our phosphopantetheine chain coming down here. You'll see that the protein makes a number of productive contacts with the coenzyme A. For instance, this arginine residue that we have over here, arginine 52, makes a salt bridge to this 3' phosphate that we have on our coenzyme A. Our phosphopantetheine terminus is capped, on the other hand, by this tryptophan residue that sits on top of the end of this phosphopantetheine chain. Okay. Now, when coenzyme A binds to the ACPS undergoes a number of significant conformational changes in order to accommodate the binding of this cofactor. So here we have, in red, we have the structure of the protein in the absence of coenzyme A, and in the purple and the blue we're showing the two subunits of the CoA bound form. Okay. Let me point out a few key points. First thing I'd like you to note is that we see a general tightening up of the structure. So for instance, these helices that we have up here move closer into the active site. Okay. We have an ordering of the structure, so where we have this loop region here, this becomes slightly more ordered, and packing in, basically allowing this binding of this 3' phosphate to occur. Okay. The other key things that we'd like to point out are these histidine and aspartate residues that undergo some conformational change and have been shown in other studies to have a mechanistic role. Now, I'm not going to go into it today, but our paper discusses the mechanistic role of these two residues and the structural biology of these two residues in particular. Over here, we're seeing a loop that we call ML3 undergoes a major conformational change. Okay, so this is adjacent to where the phosphopantetheine chain is going to bind. Okay, ML3 is pushed down and away from where it's in the normal coenzyme A unbound form. Okay, so we're seeing these major structural changes as a result of binding this substrate. That's not unusual. Many enzymes will undergo structural changes in response to the binding of a ligand or a substrate. Often it's to bring catalytic residues into a more productive position. Okay, let's have a look at this binding of the phosphodiester to this magnesium ion here. Okay, so the coordination sphere of this magnesium binds a water and aspartate, but particularly the phosphates of this coenzyme A. Remember that this phosphate is going to be, this bond here between these two phosphates is going to be broken by ACPS, and it's going to be transferred, the phosphopantetheine is going to be transferred onto the ACP. Okay, so this chelation to the magnesium, this binding to the magnesium, is locking this coenzyme A into the right conformation so that we can attach our ACP to the phosphopantetheine, but it's also serving to activate this phosphodiester in order to make that reaction easier. Okay, now we talked a little bit at the start about how coenzyme A's, ACPS can use a wide range of coenzyme A's. In this paper we've co-crystallized ACPS along with coenzyme A and acetyl-CoA. Okay, now this structure shows an overlay of those. Okay, and what you'll be able to see, here's ML3, which, if you remember, that's the loop that gets pushed out of position when we bind coenzyme A. Okay, now that shows very little structural change when we bind this larger acetyl-CoA group, acetyl group. Okay, if you look at the phosphopantetheine chain as well, that doesn't undergo a huge amount of conformational change. We see some conformational change around the magnesium, position, the magnesium gets moved out of position, as does the phosphodiester backbone. But in general, this structure looks very similar to when it's bound to coenzyme A. So what's going on? What we've done in this figure is docked, so use a computer model to model in acyl carrier protein onto the surface of the ACPS. And we've got two models presented here. The red space-filling model at the bottom is when we have no coenzyme A bound. Okay? And when we have no coenzyme A bound, you'll see that basically the ACP juts up against this ML3 loop. Okay? As soon as we bind coenzyme A though, ML3 moves out the way. And if you can imagine this red bit not being there anymore, you now have a much larger pocket in which to place anything that's attached to the ends of the coenzyme A. Okay? And it's this formation of a large open groove or pocket that allows it to accommodate the wide variety of substrates that we can use with ACPS. Okay. So that explains why the coenzyme A specificity is so broad. Let's have a look at our ACP binding. Now what we've done in this paper is, unfortunately, we've not been able to co-crystallize the ACP with the ACPS. We did try, unfortunately we weren't able to manage that. Now Paris et al. Had managed to co-crystallize the B subtilis ACPS along with ACP. And we've used that effectively to model in our ACP as well. Now Paris highlighted a number of contacts which he proposed as being forming the interface between the ACP and the ACPS. Okay? So the ACP in this figure is shown in yellow, and the ACPS trimer sitting here. You'll find that the ACP sits in the groove in between these two of these subunits of ACPS. Okay? So our active site is sitting across two different subunits. Okay. We've mutated residues, so 53, 47 and 41 are the key ones that I'll talk about, but we have mutated others to find out what they do. Okay. All of them reduce ACP binding, but aspartate 41, which is adjacent to where we're attaching the serine, and it goes right into that active site there, mutagenesis of this significantly impairs our ability of the ACP to bind to the ACPS. Okay? So we get an idea that we're having this conserved residues up and down this helix, which lock our position in place. But if this aspartate is in the wrong position, aspartate 41 is in the wrong position or is absent, then we're not going to put our serine in the right position so that we can transfer the phosphopantetheine onto it. Now where we've extended this work is to have a look at not just the natural substrate, which would be the fast ACP, but also look at a polyketide synthase ACP. When we mutate the same residues on the polyketide synthase ACP, what we find is that they also reduce and have the same effect on the binding affinity as the fatty acid synthase ACP mutants do. Okay. So it appears that although we've got two different ACPs, they're using the same recognition motif in order to bind to ACPS. Now, studies of ACP binding to other enzymes in fatty acid and polyketide biosynthesis have shown that by far the most conserved interface are these helix 2 glutamic acid residues. Okay, they act as a negatively charged groups, and these are recognized by positively charged groups, either in this case arginine residues, but also you'll find the lysine residues being used. Okay, let's have a look at a little bit more detail. Okay, this is from our model. We have a pairing up of our positively charged arginine residues and negatively charged aspartates and glutamates, and they hold the whole complex together. Okay, just that electrostatic interaction between the two. Okay. Now these residues are conserved amongst ACPs. Evolution has conserved the ability of ACP to recognize its partner proteins using this interface. And that explains our broad substrate specificity that we have with ACPS. ACPS is merely looking for an ACP that is presenting the same series of negatively charged residues. And so long as that ACP presents those negatively charged residues, it's able to transfer it onto it. That explains why we can take the Streptomyces coelicolor ACPS and use it to phosphopantethienylate a wide range of different ACPs. Okay. In conclusion, hopefully you've seen that we've done a lot of work to understand the substrate specificity of Streptomyces coelicolor ACPS. You'll see that it's both broad and substrate in terms of the coenzyme A's that it will work with, but also in terms of its ACP. And by using a combination of structural biology and biochemistry, we've been able to probe the function of this important enzyme. Thank you.